So Anna gave you a brief update earlier on the RUSP nomination application that we had for Krabbe disease, and she did show a picture of the group of us that were on the writing committee, and I'd like to acknowledge them again. It was Dr. Maria Escolar, Dr. Joanne Kurtzberg, Dr. Dieter Matern, sorry Dieter, Dr. Barbara Burton, Dr. Michael Gelb, David Wanger, and myself, and we basically took the nomination and divided it into where each of us could really answer each of the points of the nomination. So let's go to the next page. Okay, so recall there were some evidence gaps that Dieter brought up in his talk. It was nominated back in 2007, and it was not added to the RUSP by a vote of 8-7, a fairly close vote at that time. There were 3 evidence gaps that were identified, and one of them was early infantile Krabbe disease and the condition, and there needed to be a consensus about a case definition of what constitutes early infantile Krabbe disease. I think some of the newborn screening that was a lot of this was based at that time solely on New York State numbers, and we were learning a lot. There were a lot of potential, what I would say, cases of Krabbe disease that under normal circumstances people would, call a Krabbe disease case. And it's interesting to me. I'll bring this up because it's sort of been brought up through the rest of the talks. For example, if you have an individual with low enzyme activity for Pompe disease and 2 mutations, that's considered a Pompe disease case. Well, with infantile Krabbe disease and the need for the kind of treatment, that was not really considered good enough. So there was a lot more concern about what was a case. And that's really what the point of the matter was. Well, how will we know what we're identifying through screening? And I think this has been a problem, an ongoing problem, and Dr. Stone will talk more about this and how we got through that in the recent publication that was Dr. Gelb passed around earlier. The next really big issue was the test for early infantile Krabbe disease really didn't differentiate between who might have severe form of disease and who may never develop disease at all. And cyclosine really is what helped with this, and Dr. Matern provided an update on that in his earlier talk. And finally, the treatment of EIKD was kind of in question. I think this is still one of the issues that maybe many people would bring up. And we're going to hear a little bit from one of the later talks on the progress that's being made in that area from Dr. Kurtzberg and Dr. Mojirin. Let's go to the next slide. This was the original task force that was put together back in 2015, and this group really has been charged by, I'd say with Anna's leadership and pulling us all together to really make sure we're answering all the questions that were provided on that previous slide, as well as continually improving newborn screening, as well as keeping people up to date on those changes that have been made and ensuring the best screening is being done throughout through all the states that we're screening at this time. So this is still an active group, and it's now considered the Krabbe Consortium, and you'll hear a little bit about that from Dr. Joanne Kurtzberg's talk coming up after this talk. Next slide, please. So these are some of the papers that have come out through, largely through the efforts of the Krabbe group shown on the previous page and others. And we're going to, like I say, hear more about these as we go along. But the early paper, or the most, I guess, helpful paper in many regards would be the cyclosine paper. And, even though New York was able to put this together early and show that cyclosine was promising, the amazing work of the Mayo lab led by Dr. Dieter Matern and his crew really was instrumental, I guess, in providing states and anybody that's going to be coming onto this with the information needed to evaluate a screen positive case when you have second-tier cyclosine testing. The next paper, which was led by Dr. Gelb, was yes. Oh, sorry. so it was Dr. Gelbin trying to make sure that any laboratories that were performing cyclosine testing would get the same results. This is an interesting approach and provides everybody with the same set of standards that have been checked and monitored over time to ensure their accuracy, and the ability to get the same numbers from laboratory to laboratory, and made it so it's possible for each program to have hopefully a really good way of assessing the cyclosine results in a uniform manner. And like I said, Dr. Stone's paper, which we'll be hearing about, and then the neuroimaging studies and MBS outcomes study will be in this session as well. Okay, just a little more on the RUSP, which will be the next page. So the RUSP application is looking for condition information and treatment, so we were able to provide that. What the laboratory tests are that are available, how it's been validated, and essentially it was kind of developed for more like pilot testing, but because there were so many states screening, we were able to use data from all of the states that are screening, and we all appreciate that information was welcome or was shared with us so freely from all the states that are screening. So thank you very much, and we got to hear their updates in the past earlier session. But all of that went into this RUSP nomination package. There's the wide availability of confirmatory testing, second tier testing, and it's something that all states should be able to take advantage of, as Mike alluded to, and the necessity for that. And then we had, as I said, the prospective population-based pilot data, which was just all the states doing this this screening now, many live for multiple years. So as Anna said, the nomination package is complete and has been submitted, and there was a letter detailing how these evidence gaps showed earlier were addressed. So now all we can do is cross our fingers and hope everybody sees the value of the screening and that it is something that's ready for prime time.